programs for the MIT Club of Rochester. And this is an event that's part of a monthly seminar series that's jointly sponsored by the clubs of Rochester and Western New York, where Western New York is Buffalo, and it's about 90 miles down the thruway. And one of the silver linings of the pandemic is that we've gotten to really know each other in each clubs very well, and we're looking forward to when we can meet live and see each other. And by having joint clubs, we're able to get bigger audiences for our speakers and actually have more people to interact with and have a lot of fun. The talk tonight is by Professor Bryan Bryson. I'm going to let Dave is going to introduce him, but before I turn it over to Dave for the introductions, just a very simple rules. First, we'll ask everybody to keep their microphone on mute unless you're going to be asking a question. When you ask a question, you can either send me a chat, in which case I can ask it, or you can unmute your mic and ask the question that way. We found that for groups of about the size, which should be about 30 when the dust settles, that we don't really need much moderation. People can ask questions when the time is appropriate. And I wanted to ask just before we started, Bryan, do you want questions? Do you want us to collect the questions, or do you want them asked during? Do you want to take pauses during your talk, or how do you want to do it?
People should feel free to type in. I'll do it like I do in class. Type your name in the chat box if you have a real-time question, and I'm happy to mute. I'm happy to pause and answer the question right then, or we can save them to the end. Or if you want, I'm happy to take questions any way they come.
Now that sounds like a good way to do it. Okay, everybody heard that. Questions, do it on chat, and then you can, then we'll handle it that way like he does in class. Okay, so with that, well, rather than waiting for more people to sign in, they'll be coming in the next few minutes, I'm sure. I'm going to turn it over to our Vice President of the Club of Rochester for the introduction. So Dave, it's all yours.
Yes, okay. Yeah, I'm Dave Ortman, and I'd like to welcome Professor Bryson. Reading a short bio, Professor Bryson is the Esther and Harold Edgerton Assistant Professor of Biological Engineering at MIT and a member of the Ragon Institute of Mass General, MIT and Harvard. Bryan completed his undergraduate studies at MIT in mechanical engineering before obtaining a PhD in biological engineering, also at MIT. Bryan started his lab in the summer of 2018, where his lab focuses on bridging quantitative approaches to understand how the immune system eliminates deadly pathogens. So I'm happy to welcome Professor Bryson for his talk in our continuing, as Bob mentioned, our continuing Tech Cafe series with the talk entitled Engineering the Host-Pathogen Interface. Professor Bryson?
Awesome, thank you. Thank you for the invitation to speak today, and thank you for the introduction. So I'm gonna go ahead and share my screen here, and then we'll just jump right in. So as Dave mentioned, my name is Bryan Bryson. I'm an assistant professor in the biological engineering department at MIT, and I'm really excited to share with you some of the work that we've been doing in my lab that I think is very timely and appropriate. But before we go on to the meat of the talk, I always like to start this with this image from my from work that I actually did when I was a postdoc. So what are you looking at? So you're looking at human macrophages. These are kind of your front line of the innate immune defense that were subsequently infected with virulent Mycobacterium tuberculosis. Now, this is an engineered strain of the bacterium where all the bacteria constitutively express a red fluorescent protein and have a drug-inducible green fluorescent protein. And so what we can do there is then at different time points, we can add a drug and then we can ask which bacteria retain the ability to turn on green fluorescent protein. And this is a marker of whether the bacteria remain kind of transcriptionally and translationally viable or alive, so to speak. And what you can see from this is in blue are our nuclei, in red are bacteria, and red and green are the ones that remain alive. And what you can see is that there's striking heterogeneity that exists in this system. And this is actually probably the name of the game when we think about host-pathogen interactions, because many times what really kind of the original sin or the inciting event between an individual host immune cell and a pathogen like MTB is the interaction between a single bacterium and a single macrophage. And that is what really kind of drives the fulminant like evolution of disease. And this is really one of the inspiring questions in our research group. And what our research group really does is we like to think about how we can bridge molecular immunology, bioengineering, and quantitative modeling to study host pathogen interactions. And on this slide, on the left here, you can see a cartoon of a virus, and on the right is a cartoon of bacteria on an agar plate. And one of the things that I hope that you can take away from today's talk is that really the theme of today's talk is that small changes can have big impacts. And we can think of this through the lens of kind of our microbial communities, viruses and pathogens that have wreaked havoc on human history. But hopefully you'll also see how I use this kind of theme as a turn of phrase in the second half of my talk to think about how we can use new approaches to understanding viral evolution. And so to talk to you about my first love. My first love is tuberculosis. Tuberculosis is one of the leading causes of infectious disease death. And as you can see, much of that disease burden is shouldered by the developing world. And just to put some numbers on the problem, tuberculosis is estimated to infect about 1/4 of the global population. So billions in the B's of people are infected latently. This amounts to about 4,000 deaths per day. Now, again, when we think about tuberculosis, there's so many different layers of heterogeneity. So let's talk about what happens at the people level. So while we estimate that about 1/4 of the global population is infected with the bacterium, only about 1 in 10 of those individuals develop what we call active TB disease, which is characterized by weight loss, fatigue, coughing up blood, et cetera, et cetera. Now, not all hope is lost because we do have an antibiotic regimen that is effective for drug-sensitive TB. But as you can see here, this is a 6 to 9 month regimen of 4 pills a day. And so you can imagine how in places where public health and distribution of these antibiotics is limited, you can imagine that this is kind of a perfect storm for the development of drug resistance. And from my perspective, what would be transformative? And I think we've seen in the last 15 months how vaccination can be transformative as it's getting us out of this COVID-19 pandemic. While we've been calling it tuberculosis for over 100 years, we still don't have an effective vaccine to protect against TB disease. So just to like put things in context, we sequenced the SARS-CoV-2 genome in December of 2019, and we now have like multiple vaccine candidates in the pipeline in arms. TB, we're still working on it. Okay, so given that, the big question is, if we want to get to a vaccine, our hypothesis in our research group is that we really need to understand what does the immune response look like, and how can we perturb the immune system to really give the response that we would desire. And in our case, this is a response that would look like something like killing all of the bacteria. All right, so just so we're all on the same page about how we think that this works, let's just kind of give you kind of the immunological life cycle of what we understand about how TB disease is initiated. So TB is transmitted via aerosol. So an infected individual will cough. This will aerosolize bacteria into droplets. Those dropletized bacteria that are inhaled by another individual, proximal. And those bacteria make their way down into the alveolar space of the lung, which is patrolled by these frontline immune cells called alveolar macrophages. And the job of this cell is, you can see it in the name, macrophage. It's really there to eat up things that are around. And so what happens is that the bacteria makes its way in, these patrolling immune cells then take up or phagocytose the bacteria, and this results in the kind of the first domino falling over, so to speak. And so this results in the kind of initiation of an innate immune response, where you'll have these infected macrophages then start secreting things to get other cells to come to the site of infection and really kind of drive the kind of evolution of disease. So then you have an infected cell, you have nearby uninfected cells that then come and kind of swarm around this kind of this infected cell. And this creates kind of what we call one of the hallmarks of TB disease, which is called a granuloma. Now, you don't need to know very much about the granuloma, other than it's in a highly dynamic kind of cellular ecosystem, so to speak. So the granuloma will then go on to mature. You'll have other immune cells arrive to the site of infection. Now, what's kind of remarkable about the granuloma is that you can imagine that there might actually be multiple granulomas in a given individual because they've been kind of repeatedly challenged with TB infection. Now, some of these granulomas will become fibrotic and hard. Some of these granulomas will become necrotic, where the cells die and the bacteria then expel into the airway. In the same individual, you can have some of these granulomas that fully sterilize, meaning that they killed off all the bacteria. And then in some granulomas, you will have bacteria that are just happily growing. Now, again, bringing it back to the concept of heterogeneity, within one individual, you can have sites of immune success where you are fully clearing the bacteria, and then other sites you are not. But what gives me hope as a now card-carrying molecular immunologist is what this suggests is that the immune system can kill bacteria. And in particular, this bacteria, which is one of human history's top 10 killers of all cases, of all causes. So what this suggests is that the immune response can do it. What we need to figure out is, what is the recipe? Now, okay, so this is what we understand about the immunological life cycle, but there are a number of unknowns that remain. Like I said, we know that the immune system can get us to control, to eliminating bacteria in particular regions. We don't know how it does it. We also don't know how the bacteria is trying to resist the mechanism, these antibacterial responses. And notably here, these knowledge gaps are fueled by several limitations. TB is not a model bacterium. It's not like E. coli that doubles every 35 minutes. TB doubles about every 22 hours. So when we do an experiment, we like plate bacteria out, and then we go on vacation and we come back 17 days later, and then we can count how many bacteria grew out of a particular experiment. So just the life cycle, just kind of the slow growth, is really challenging when you think about trying to develop kind of an intuition or a model for how the immune system responds. And the other thing that I'll say is that, I, maybe as an engineer, TB kind of calls to me because of the following, because there is no kit. There is no kit to study TB. Everything that we do, we have to build ourselves. All of the methods and tools we have for TB are kind of homegrown solutions, which maybe this is why as an engineer, I'm drawn to the problem, because I get to tinker and build and do those types of exercises. Okay, so this was a question that really inspired my research group when I started it. And you can imagine that this, the question of what does the early immune response to TB look like might be something that many people have already answered. But I made the argument that I think that many times we have maybe made the wrong assumptions for how we might think about modeling these kind of early immunological events, these transmission events in the laboratory setting. Now, why is that? So in order to kind of justify why I think that we've kind of missed the mark in some of our early experiments with TB, we have to first go to the non-human primate model of tuberculosis. And why is this model attractive? Because the non-human primate model of tuberculosis is actually probably the animal model that best reflects kind of the clinical spectrum of disease that you observe in humans. And what did they do? So using kind of genetically barcoded strains where each bacteria carries its own unique kind of like barcode, what they did is they can do a low dose infection in the lung where they directly install bacteria in the lung, and then they can allow disease to progress, and they can use PET-CT imaging as you would if you were going to the hospital, and they can identify where there are regions of kind of high inflammation in the lung. And that is kind of indicative of where TB disease is. And then at the time of kind of doing deeper analyses of the animal at time of necropsy, they can then harvest these individual lesions and ask how many unique barcodes are present in any one of these granulomas. And what you can see here on the right is, by and large, these individual granulomas are founded by a single barcode. And so what does this mean? This really, again, really emphasizes this idea that kind of the initial kind of initiating event in TB infection is the interaction between a single bacterium and a single macrophage. Okay, so that kind of blows my mind. All right, so this is really exciting because it says that very few bacteria can establish infection. But the real question here is this what's happening in real life? And so to answer that question, we'll go now to South Africa, where what was done is they were built this really kind of engineering marvel, I'll call it, where they ask an individual upon active TB diagnosis to actually sit in this small room. The small room has a chair, it has a TV, and they're just like asked to just like breathe and operate normally. And what's really exciting about this room is that it's also, as you're kind of expelling air and just kind of breathing normally, what the room is trying to do is trying to evacuate all of that air that has been expelled into a kind of a concentrated solution, such that you can then subsequently come in with fluorescent probes to try to identify and enumerate how many bacteria were expelled in that period of time. Now, does anybody want to take a guess for how many bacteria we hypothesize are expelled by an individual with active TB disease in the span of an hour? Does anybody want to take a guess in the chat box? Okay, 300. All right, 300 is a guess. Okay, anybody want to go higher or lower?
Here, wait just a second.
Okay, great. All right, so we've got some highs, some lows. Actually, when they actually went to enumerate how many bacteria are expelled by an individual with active TB disease, it's on the order of tens of bacteria are being expelled, which is kind of mind-blowing because, you know, that's not a lot of bacteria. And so a real question is, for us, is how can the immune system really, is the immune system equipped to really respond to a single bacterium? Because you can imagine that, you know, it's kind of like the story of the boy who cried wolf, where you kind of only want to cry wolf when there's a real problem. And so you can imagine just in the span of like kind of normal activity, we're being bombarded, our immune system is being bombarded with lots of foreign objects, be those pathogens or just foreign debris. And a question is the immune system always going into inflammatory panic mode? Or are we instead restrained in terms of thinking about how much signal or how many bacteria are required to really go into alert? And so again, returning to this question of what does the early immune response to TB look like, we're inspired by this beautiful conceptual model, an experimental model that was done by some people at the NIH. And so now here we're really gonna get into the biology of it all. And so what we're looking at here, now we're looking at human or mouse immune cells, mouse macrophages in particular. And what they were done is they were challenged with kind of a chemical, with a component of a bacterial cell wall, which is very bioactive. And then they incubated this with these immune cells and then asked about the ability for these immune cells to kind of mount an inflammatory response. And what's really impressive about these techniques that we use these days is that we can measure this not for kind of an ensemble measurement, but we can measure this for each individual cell. And that's what we're looking at here using these techniques called flow cytometry, where on the y-axis, you can say, for example, look at the production of one individual inflammatory protein. And on the x-axis, we look at a different inflammatory protein. And so what was done here is just a simple dose response where you escalate the amount of bacterial cell wall component that you treat. And you can see here in the absence of stimulation, these cells are not producing these inflammatory proteins. Now at the other end of the spectrum at 100 nanomolar, so very high concentration, nearly all the cells are responding and producing both CCL3 and TNF. But what you can see in the kind of the intervening concentrations is that it's almost like it's a very, to go back to something we probably all learned about in like 1801 or 2, it's a sigmoidal response. It's ultra sensitive and it's switch-like, which is really remarkable, suggesting that the immune system tries to filter out kind of low kind of blips on the radar. And then it really only goes into full-blown activation mode once we kind of tip the scale. All right, so a question then becomes is how can the immune response see a single pathogen? Because we know that TB can cause full-blown disease with a single pathogen. And so is the immune system equipped to do that? All right, so this is work all done by a fantastically talented graduate student in my group. And one of the things that's really important as I described that previous experiment is that TB does not really operate the same way that all these many of the pathogens that we've studied, especially bacterial pathogens. So TB is called mycobacteria because it has what's called a very rich mycolic acid on the cell wall, which is really just think about this bacterium as like wrapped in a ball of wax. And as a consequence, it really doesn't tend to interact with a particular set of receptors. And what you need to know here is that TB, because of its kind of complex cell wall, interacts with a number of different receptors on the surface of immune cells. And so what Sydney first wanted to ask is if we go in with a purified ligand for one of those receptors that we know that TB interacts with, do we see this kind of similar ultra-sensitive response? And the short answer is yes. And so what you can see here is now on the y-axis, we're just looking at kind of a feature of the cell shape. And on the x-axis, we're looking at TNF-alpha, again, this inflammatory protein. And what you can see here is as we can kind of increase the concentration of this ligand, we see this kind of, again, small ultra-sensitive response where at like a nanogram per ml, you see like just a small population of the cells responding. And then when you go up to really high concentrations, one of the things that was really interesting is that as you can see at this really high concentration, it actually doesn't go to 100. And yes, we could go higher, and we actually did go higher. And one of the things that was really interesting was what we then found. So now I'm just taking that same data and I'm just plotting kind of summary. And so this is just the quantity of cells that were actually bona fide expressing this protein. And then on the x-axis again is concentration. And again, you can see the switch-like response. But one of the things that was really interesting in our observations is that even when you go to really high concentrations, the system does not go to 100% response. We cannot get all of the cells to respond. And this is kind of mind-blowing because it suggests that even if you were like putting as much signal into the system as possible, maybe 1 and 2 of the cells would actually respond. So when you think about this in the context of an intact pathogen potentially landing in the cells, you can think about this as hitting the lottery. You're either going to hit a cell that can respond to you or a cell that can't respond to you. All right, so now the real question is this is the hypothesis that maybe there's this kind of lottery-based system for whether an immune cell can respond to an individual bacterium. And so we wanted to ask how this conceptual paradigm would actually translate to intact bacteria. Because a lot of people do these experiments where they take kind of like a component of a bacterial cell wall and like treat cells with it, but that's not what kills people. Like what kills people is an intact bacterium, and what we'd like to understand is what is the immune response to an intact bacterium. All right, so what's challenging about this? So what's challenging about this is actually that when we try to infect immune cells with very few bacteria, it follows a Poisson process. So if you add 50,000 bacteria and 50,000 immune cells, what happens is that probably only about 30% of the immune cells actually end up getting infected with bacteria. And so if you're trying to just look at that kind of response en masse, you would have 70% of your cells that were uninfected and 30% of your cells that were infected. And so what we reasoned is that if we really wanted to understand what was happening in the cells that were directly infected, we needed to use a slightly different strategy where instead we could actually fluorescently label the bacteria with a green fluorescent protein and then look at the production of a particular inflammatory protein in a different color. So then we could look for those cells that were truly infected with bacteria and look at the quality of the immune response in those cells. And so that's what we'll call bystanders. So these are kind of onlookers. These are cells that are not directly infected. And then we'll have these cells that are in fact GFP positive as the truly infected cells. Okay, so the goal here is really to have as precise of a measurement of the immune response in directly infected cells as possible. All right, so what did we find? So now on our y-axis, we're looking at TNF-alpha, this inflammatory protein, and on our x-axis, we're looking at GFP. And really, you just really need to focus on this MOI of 1, which is multiplicity of infections, so 50,000 bacteria, 50,000 immune cells. And what you can see here is that there's a population of cells that don't that aren't infected, a population of cells that are infected with, that have GFP expression. But then when we look at how many of the cells that are infected with bacteria are producing the inflammatory protein, 1 in 50. 1 in 50. So you can see 1% of the cells out of this total kind of right half, of, which are the GFP positive cells, 1 in 50 cells is actually responding by producing this inflammatory protein, which is kind of mind blowing because it's like, again, this kind of lottery. It's like you're an infected cell and you're kind of getting away, and like the immune response is not even mounting an inflammatory response to you. Okay, so, you know, the real challenge that I think about when I think about host pathogen interactions is that you have a bacterium that has its own machinery for survival competing against an immune cell which is trying to eliminate you. And so the question is when we see this response that only 1 in 50 cells is responding, there's actually a few models by which we can interpret this data. One is that we could argue that TB is a weak agonist. It does not turn the system on really well. Or we could argue that TB is really clever and it's just undermining this process. It has evolved with humans for a very long time, and as a consequence could just be evading the immune response. And to maybe make a long story short, we did a whole host of experiments to try to test between these two models. And what I can tell you is that the following. TB does not appear to be like an exquisite manipulator because protein synthesis is going by is occurring normally in these cells that are infected with TB. So it's not that TB is disrupting the ability for an immune cell to produce protein. It's also not directly inhibiting the ability of this inflammatory protein to be produced. So our hypothesis is that, again, TB is likely a weak agonist. Now, for those of you who recall freshman biology, you know, it's the way in which a cell responds to its environment is incredibly complex because we have these sensors or receptors on the surface of the cell that really kind of survey and patrol the environment. And they're kind of always sampling to see what's in the environment. And then it's taking these signals that it receives kind of extracellularly and transmitting that to the immune cell to make the immune cell mount an immune response. And so we have this receptor here. Toll-like receptor 2. You don't need to know anything about it other than it's a receptor that can survey the environment. And then when it binds to something that it is binds to its ligand, it transmits this really complex cascade of signals to the nucleus of the cell, turning on the production of inflammatory genes such as this TNF alpha that I was describing to you before. Now, it's a more complex input-output relationship because there's all these really complex biochemical signaling events that are occurring. And what we know about the system is that these particular proteins, for example, this transcription factor, which drives the transcription or production of RNAs, is important for inflammatory protein production, as well as these other enzymes called the MAP kinases. Now, what was really exciting for us is we could actually similarly use highly sensitive techniques to begin to ask the question of like, what's happening with these proteins and their activation state? And to maybe make a long story short, we start to look at these 3 proteins initially, I-kappa B alpha, ERK, and P38, really with the idea of trying to say, okay, what happens, what's different about TB from one of these soluble ligands that we just kind of normally like just treat cells with. And so we did a whole host of experiments. And one of the things that was really exciting when we did those experiments is we found the following. So if we looked at a particular protein, P38, and I'll tell you a little bit more about it in a second, we can see that when you add a soluble ligand that's just kind of a synthetic ligand for this receptor, we see this really robust activation. So we see this kind of time, like P38, its kind of activation level increases acutely and then it kind of decreases. But when we add TB it's silent. Nothing is happening with this protein in response to TB. Now, what do you need to know about P38 for this talk? Really, P38 is a master regulator of both how immune cells die, but also how immune cells make inflammatory proteins. So the fact that we're seeing that that P38 is not getting activated is important, because if it's not being activated, it's likely not also going to be allowing for these inflammatory proteins to be produced. So this kind of gives us an idea of where in that system are we seeing these bottlenecks in kind of activation. Okay, so that's just a little flavor of how we're using highly sensitive techniques to really try to probe in individual cells how the immune system kind of responds to infection. But I framed it all around this question of the immunological life cycle of TB. So how in the world can we argue that we're doing these experiments in tissue culture dishes and an incubator when TB is really incubating in humans? And we'd like to be able to understand how we use it, kind of our experimental lab-based models, to reflect what's happening in humans. Now, I'm going to make the argument that what we're observing in our kind of cell culture models in the lab are actually very reflective of what we're seeing in people. And I'm seeing a question in the chat box about P38 blocking inflammation. So P38 is actually like a second key. So P38 actually unlocks the ability for for inflammatory protein production to occur. So if P38 doesn't get turned on, our hypothesis is you're not going to get the rest of the inflammatory response to turn on an inflammatory response. And I see another question from Brian about, are there two distinct classes of host cell, uninfected? So that's a really good question. So I would say that it's very much a Poisson process. So if you go ahead and you take those bystanders and then re-challenge them, a subpopulation of them will be infected. So I don't think that they're resistant to taking up bacteria. I just think it's really a luck of the draw thing. But great question. Okay. So I'm going to argue that, okay, what we're observing in the lab is actually reflective of what we're seeing in animals or people. So how will I make that conclusion? So actually, if you infect, again, non-human primates, which are the best kind of animal model of human TB, and then you just look in the blood and ask how many genes are kind of turned on or just differentially expressed following TB infection, what you find is that it takes about 10 days to get 500 genes differentially expressed, keeping in mind that there are 20,000 genes. So that means you've infected an animal with TB, and it takes like a week or more than a week to really get a robust kind of gene expression change in response to TB infection. Okay, if I were being super critical of what I just said, I would make the argument that, okay, Brian, you're infecting in the lung and you're making these conclusions about what you can measure in the blood. And obviously we measure things in the blood often because it's an incredibly accessible compartment. Now, what if we could actually go to the lung specifically and ask about lung responses in response to mycobacterial infection? So the dirty little secret about the TB field is that we do have a vaccine called BCG. It's kind of an attenuated cattle mycobacterial strain. It actually doesn't work very well in its kind of current mode of delivery. And so we've discontinued its use in the US for TB. But in South Africa, it's still widely used. And so what they reasoned is that people who had been previously vaccinated with BCG could volunteer for a study, come back, and then be challenged with BCG in the lung. And then you could ask, like, how many bacteria were needed to mount or generate an immune response. So they started with 100 bacteria, and they said how many genes or proteins were being differentially, the levels were different, and if you compare uninfected to infected. And they didn't find very much when they added 100. So then they said, We went up to 1,000. We still didn't see anything. So then they said they went up to 10,000 and then they finally saw things. But I hope you can realize that 10,000 is not really reflective of what the true kind of like bacterial infection event looks like, given these new studies about how many bacteria are actually being expelled by an individual with tuberculosis. So hopefully in this first half of the talk, what you've been able to get a sense of is how we're trying to make use of kind of ultra-sensitive techniques to really try to monitor how individual bacteria kind of catalyze in these immunological events in cells, and how we can then use that information to begin to think about where do we need to intervene with small molecules or vaccination to try to modulate the immune response to infection. So now I'm going to switch gears a little bit and just give you a different take on how we as MIT engineers are trying to understand host pathogen interactions. And it's not lost on me that life occurs on Zoom because of the COVID-19 pandemic, which is one of the kind of worst pandemics in recent memory. And as of today, the number of cases just kind of blows my mind. And so, as a junior faculty, it's actually kind of terrifying when this happens, because you finally feel like, okay, yes, I know how my lab works. We're like 2 years in, like, cool, we have it all figured out. And then you get told that it's time for you to shut down. And you just finally feel like you've hit your swing, and then everything just ends. And all those experiments that you had planned just kind of, they're not happening. And so one of the things that I told my lab quickly that we needed to think about is how could we utilize public data that are just openly available through the NIH to kind of devise new strategies or devise new hypotheses, because who knows when we're going to go back to lab. And honestly, we're back in lab, but we're still not at 100% capacity. So a question that we wanted to ask is, what can we learn from publicly available data? Is there some experiment that somebody, or some analysis that somebody did not do previously on their data that we could instead do and generate a novel set of insights? And so we were really actually inspired by a class of algorithms because of this student, Bryan He, who's now a Stanford Science Fellow. And what you need to know about Bryan He is that he was an amazing algorithmicist, but also has a deep appreciation of literature. And actually, when he was applying to PhD programs, he was deciding between Harvard literature or MIT electrical engineering computer science. And he was really inspired by a class of algorithms called natural language processing algorithms. And what you need to know about these algorithms is what they do is they automatically extract patterns from big data sets. And if you think about sending an email via Gmail, and now Gmail is predicting the next word, or if you're sending a text and your iPhone is making a prediction about the next word, these are powered by natural language processing algorithms. And specifically, they're kind of powered by this concept of distributional semantics, which is a concept introduced in linguistics in the 1950s. And the idea is that you shall know a word by the company it keeps. So by looking at the relationship between words and their position and how they co-occur, you can really begin to kind of develop a kind of a probability distribution for how words are going to co-occur. And specifically what these algorithms do is they learn the probability of a word given its context. So you can take the word or the phrase, the Chinese president blanked to Japan yesterday, and you can imagine that the word went has a 50% chance of filling in this blank. And so this is what natural language processing algorithms do. And you're probably thinking to yourself, what in the world is he talking about natural language processing in a talk about host pathogen interactions? But we'll get there, I promise. And the idea, kind of coming back to this theme that I introduced earlier, is that small changes can have big effects. And you can imagine small changes having big semantic effects. And so we can start with the sentence, the boy pats the dog. If we change the original A to an E, the sentence becomes the boy pets the dog. And that sentence, so you can imagine this is still a grammatical sentence. However, the semantic meaning is not that different. Now, by contrast, if we change that original P to an E, the sentence becomes the boy eats the dog. Now, this is still a grammatical sentence, but it's a fundamentally different meaning than the original. And so we were really inspired by this idea that when we think about language, we can think about grammar, but we can also think about semantic effects. And so what we reasoned is that we could initiate this kind of algorithmic framework called constrained semantic change search, which is a sequence of tokens from some language. What you want to do is you want to find the single token change that, one, induces the largest semantic change, and two, is constrained by the rules or grammar of that language. So instead of just optimizing for grammar, what you also want to optimize is for semantic change. You want to use them both together. And so you can imagine that that works really well for a sequence of words from the English language, but you could also think about the language of proteins. A sequence of amino acids from a viral protein. And that's the idea that we pursued, where if we think about, if I'm a virus and I'm trying to, if I'm evolving and I'm trying to avoid the immune system, what I want to do is I want to evolve in such a way that I preserve my key functions. I need to still retain the ability to infect human cells, but I need to look different to the immune system. So I want to undergo some amount of protein language semantic change. All right, and so maybe I'll skip this slide, or maybe for those of you who are course 6, we're using state-of-the-art neural network frameworks to really apply these techniques. So how does this actually work? So if we actually ask the algorithm to kind of perform constrained semantic change search, what happens? So we can take the original news headline, wine growers revel in good season, and we can apply constrained semantic change search. We can ask it to make a grammatical single word substitution to the sentence that is the semantically closest to the original headline. And what it actually proposes is wine growers revel in strong season. So that feels kind of intellectually fulfilling. Now we can ask it to make the single word change that makes the semantic meaning as different as possible to the original headline. And it actually proposes wine growers revel in flu season. I just find that appropriate as we're thinking about infectious diseases. Okay, so again, the idea here is that small changes can have big impacts. And our hypothesis is that the same way that we could learn the language of English by training on available kind of corpuses of English language text or news headlines, we can instead go to the language of proteins, which is amino acids. And each one of these amino acids we could take as kind of a training data set to learn the language of a viral protein. Again, because if we think about a viral protein, this is just the flu, one of the influenza hemagglutinin. And what's important to know here is that a single amino acid mutation, this histidine to serine mutation, means that this antibody can no longer bind. And so as we think about COVID, SARS-CoV-2, and all these variants of concern that people are talking about, they're variants of concern because we're concerned about mutation to the viral proteins that would eliminate the ability for an antibody to bind to that particular region. And this is really important. And so again, what we wanted to ask is, could we model viral escape as semantic change? And the first question you have to ask is, okay, we're just training on publicly available sequences. Importantly, what did we do? Again, we wanted to use publicly available data. We trained our model on just sequence data alone, no other information. So it was just given like 45,000 sequences of influenza HA proteins. And we wanted to ask, okay, what does the language model learn for influenza proteins using this framework? And what was really surprising is that, what these language models truly end up outputting is just kind of a vector of numbers that represents the semantic representation of any amino acid sequence. But we can actually take that data and cluster it and project it down into 2 dimensions and then ask, is there any enriched metadata in any of these clusters? And what was really surprising and remarkable was that when we actually looked at these individual clusters and then superimposed publicly available metadata, what we found is that these clusters were very much enriched for influenza subtypes. And what was encouraging for us here is that what this suggested is that even though we were only training on sequence data alone, not passing any metadata, what it suggests is that the language model is actually able to learn something that has biological meaning. So this is really exciting for us. And so the next question we wanted to ask is, could we actually model viral escape with constrained semantic change search? And the idea here is that for a good escape mutation, what we wanted is a mutation that affects the overall structure and fitness of the virus as opposed to something that's functionally neutral, so high semantic change. And two, we wanted something that follows the grammatical rules, something that still allows the virus or the viral protein to kind of retain the key functions. And so there, what we wanted is a mutation that does not harm viability or infectivity. So we still need to be able to infect cells if we want to survive. So again, this constrained semantic change search, because we're kind of exploring this kind of complex landscape where we can't mutate too far, because if you mutate too far, then you're not going to survive. All right, so how did we do this? So we actually modeled viral escape using our language model, where again, what we did is we used publicly available sequences that are just available in this database called the FluMap. There's about 45,000 sequences that we built a language model using. And then what we did is we actually basically performed evolution in silico, where we made each potential amino acid mutation to the influenza protein, a particular flu strain from Perth in 2009, where we made basically every potential amino acid mutation. So we took every position and mutated it to all other 19 amino acids. So we're basically creating a library of mutant viruses on the computer. And then we asked the algorithm to quantify semantic change and grammaticality relative to kind of the original sequence. And why did we use this virus, this influenza strain in particular? Because actually a group in Seattle at the Fred Hutch had actually already done the wet lab version of this experiment. So even though we could not go to lab, somebody else in the past in 2018 went to lab, did these experiments, and we could ask about our model's predictions according to what they were able to observe in their data. And what these experiments were done, were basically this lab made every potential, again, single amino acid mutation of the influenza virus that infected cells, either in the presence of antibody or in the absence of antibody. And what these experiments allow you to do is they allow you to identify the viral mutants that retain the ability to infect cells, but also the viral mutants that retain the ability to infect cells, even if you're throwing antibody on them. So these are the mutants that you really care. These are the ones that are escaping immunity. And then you can sequence them and figure out which were the ones that survive. So our hypothesis was, using our language model, that high semantic change and high grammaticality were going to be the mutants that would escape the immune system. And in fact, when we tested that hypothesis, it turned out that we were actually right. So let me just tell you what you're looking at here. So we can quantify semantic change relative to an original sequence, and we can quantify grammaticality of any particular amino acid mutation. And what you can see here is there's this upper right-hand quadrant of high grammaticality and high semantic change has all these red X's. And what are these red X's? These red X's are experimentally confirmed escape mutations. So what this suggests is that our model, trained on sequence data alone, can predict viral escape, which is really impressive and amazing. Now, the question is, all right, so can you take this one step further? Does the knowledge that we gain provide any information about how we could, say, for example, develop a universal flu vaccine? And I'll say the answer here again is yes. So what we can do is, even though we did not train our model on structure, we can actually, because the structures are available for this influenza protein, we can actually superimpose our model's predictions on the viral protein. And what you can see here is in the head domain of the influenza H1 protein, there's this kind of yellow-green color, which means there's high escape enrichment, which is like, this is a region of the protein that is just prone to likely escape. Whereas there's escape depletion in what's called the stock domain. And actually about 3 weeks ago, a research group, again in Seattle, demonstrated using a new vaccination strategy that if you actually are able to generate antibodies that recognize this part of the protein, the stalk domain, it is broadly neutralizing, meaning that it could serve as a platform for a universal flu vaccine. So I will celebrate the student who did this work because he's not a virologist. He's definitely not an influenza person. He's become an influenza person, but just by his kind of language model approach, he was actually able to recapture kind of expert knowledge of influenza, all in the span of 3 months. So a question that you might ask now is, okay, did you get lucky with influenza H1, or is this a kind of a general framework that could be applied across viruses? And maybe just really briefly, I'll say that it is a generalizable framework. So this works for influenza H3, it works for HIV envelope, and it also works for SARS-CoV-2. So what I hope to have shown you in the second half of the talk is that this kind of modeling framework and a good analogy can go a very long way. In particular, that by using these kind of algorithmic frameworks, we've been able to both kind of have something to do during the pandemic when we're locked down, but also make really important contributions to how we understand viral evolution and escape. So before I take any more questions, I just always like to give people about a teaser trailer to what is coming up next. So, you know, I think as a junior faculty, it's your dream for something you do in the lab to actually make its way out of the lab. I'm happy to share with you all that our framework of constrained semantic change search has actually been adopted by the CDC. So it's part of the genomic surveillance programs. So as a new sequence comes in, we can actually quantify these semantic change grammaticality in real time. And what this allows us to do is it allows us to kind of establish experimental priorities, because we can't test every mutant all at once. So we have to make decisions about how we're going to prioritize what mutations we make in the lab to kind of pseudoviruses to try to understand evolution in that setting. And then, you know, we're also really excited about taking these kind of algorithmic frameworks and taking them one step further in not only applying them to viral pathogens, but going back to my first love that I introduced to you at the beginning and asking if we can apply this to understanding the development of antibiotic resistance. So with that, maybe I will close, but not before I thank the superheroes of the story. So the second half of the talk was all done by a really talented former graduate student, Bryan He, who's now a Stanford Science Fellow. And all the work of the first half of the talk was done by a really talented graduate student, Sydney Solomon. I am deeply grateful for the lab that I get to work with. They are amazing people. My kind of village of really fantastic collaborators. Notably, I want to acknowledge my friend and collaborator, Bonnie Berger, in the math department, my funding sources, and you for both the invitation and the attention. And I'm happy to take any other questions that you might have.
Good. We'll wait and see whether whether anybody what goes on in chat. I've got a couple of questions, and I'm not a very good typer, so I'll ask in a minute. Let's see whether anybody else has questions first. By the way, thank you. That was really a great talk. I learned quite a bit.
This is Dave. I have an observation first. I learned a new term from your talk. I've heard of in vitro and in vivo, and I believe you used the term in silico. And I can imagine what that means. That's a simulation, but I had not heard the term before.
Oh yeah, yeah. So in silico is basically probably a third to a half of what we do in our lab, which is the kind of computational modeling of our data to try to develop some predictions. Because this day and age, it's much cheaper to run some code than it is to buy any reagents. With the plastics shortage, with the supply chain disruption in the US right now, like getting pipette tips is you have to go to eBay. You can't, otherwise you wait for 5 months for them to come from a company. So yeah, so we've become a much more in silico than in vitro lab.
Right. But here's a question, and this is from a non-biologist. Can you broadly distinguish between the human immune system response to bacterial pathogens versus viral pathogens? Is this really the same thing, or is the mechanism wholly different?
So that's a really great question. So receptors is kind of the language of recognition when we think about biology. And so for example, bacteria have lipoproteins. So like lipidate, like proteins that have some like fat chain on it. And that's really unique to bacteria. And as a consequence, there are particular receptors that actually bind these lipoproteins. Now, there's some really interesting intersections of other receptors that, for example, bind foreign DNA or foreign RNA. And you can imagine that viral and bacterial pathogens both can have both either foreign DNA or foreign RNA. So there are receptors that are shared for recognition of either bacteria or viruses, but then there are unique receptors that also can kind of exclusively recognize one or the other. And people have shown that like these different kind of recognition events definitely drive very distinct immune responses.
Okay, thank you.
I think Bryan has a question.
So how large of a training model is required? So that's a good question. I cannot tell you how many, it's very empirical at present is what I'll tell you. So for influenza, we had about 45,000 sequences. For HIV, we had 55,000 sequences. For SARS-CoV-2, when we started the work, we actually had 900 sequences. So we use a modeling framework that tends to work well in the absence of a lot of training data. So that was kind of one thing we had going for us. And so right now in working with TB is actually a pathogen that does not mutate very quickly. And so there we don't have as many diverse sequences to train with. So for some of the antibiotic resistance targets that we're looking at, we might only have 100 unique sequences, but we're trying anyways. So what I can say right now is that we don't have kind of a hard and fast rule about how many sequences are needed. But we're kind of starting to try to quantify like what is the kind of the sequence diversity that is needed as input to kind of give good kind of training results, so to speak, or good model result, prediction result. And then I also see a question from Rob in the chat box. Can the immunogenicity of TB somehow be increased to create an enhanced initial immune response? So that's a good question. So I think that where we have opportunity to enhance is like we can enhance the immune response, and maybe I'll show a slide. Let's see if I have it. Okay, cool. Yeah, I have this slide. So I'll show you what I think we can do. I don't think that we'll necessarily be able to mutate the pathogen in some way because it's restricted, or like, we're trying, I guess what I'll say is we'll have more luck, I think, modulating the immune system. And I'll just show you one slide for why I think that is the case. So here's that kind of response that I showed you prior in purple, this kind of switch-like response where we only get about 30% of the immune cells to respond. Now, if we just kind of precondition the immune cells with kind of an inflammatory protein prior to stimulating them, then we get a more homogeneous response and we get more of the cells responding. So this is actually what we think vaccination is doing, is vaccination in the setting of TB is making the population making the population response to infection more homogeneous. And so now what we're trying to understand is how can we take advantage of this kind of observation and think about what are the immune responses that we need to homogenize such that we can bias a more robust immune response. That's a really interesting question about coding it with something. We actually are working on a vaccine right now that is a coded vaccine. And so the idea actually is definitively to put more immunostimulatory things on the surface, because TB being a ball of wax is not, you can imagine that the intracellular contents of the bacterium probably aren't often accessible. So we have to put the inside out. So yeah, so we're definitely actually working on an inside out vaccine right now. And then I see a question from Bob, considering the stable stock region of the pathogen, is this the region to attack to make a more effective vaccine? Yep, exactly. So we want to prioritize regions of escape depletion, because for some reason, it does not appear that the virus can, or our model would predict that the virus cannot tolerate a lot of mutation in that region of the protein. And what we know about viral fusion mechanisms, so how viruses kind of fuse with the cells that they're infecting, is that actually in that stalk domain is a spring-loaded mechanism by which viral fusion occurs. So if you kind of do any mutation there, basically the virus can no longer fuse to cells. So it's kind of intellectually fulfilling that that is what the model is predicting, because we know, I don't want to call myself a virologist, that'd be pushing it too far, but the virology field knows that these spring-loaded mechanisms for viral fusion are very important for viral kind of infectivity. Okay, and then Laurel has a question. Could we look at the last data slide from your talk? I'm curious if the relatively small number of red crosses means SARS-CoV-2 is tougher for vaccines. Good question. Let's go back there, and I'll tell you the dirty secret of this slide. So I'll tell you the backstory of this whole paper. So the graduate student who was doing the work had this idea probably in the end of March of 2020, and he coded it up in a week. And we were working with whatever data was publicly available at the time. So the number of known escape mutations to SARS-CoV-2 that were publicly available for us to know was actually fairly few when we published the study, or when we actually submitted the paper. And so there's actually many more red X's that are now available. So as we've kind of retrained the model, we actually retrain the model every 2 weeks for SARS-CoV-2. So it's not so the relative few X's here was really kind of what data is reflective of how much data we had accessible to us at the time. But I say when you publish what I'll call this a paper, a good paper, people come out of the woodwork with extra data for you. So yeah, so we actually have a lot more data now that is that just kind of gives us some more insight. And actually, I don't think that it's tougher. In fact, what we've actually shown is that, similar to the kind of the structural arguments that we could make for influenza, we've actually found regions of SARS-CoV-2 that are depleted for escape potential. And in fact, people are already making vaccines against that region of the protein. So we feel pretty encouraged that that the vaccines will work, and that our modeling framework can help identify regions of the virus to target.
I was typing out a question, but it's easier to ask it. In terms of escape, I realized that it probably, whether you get a vaccination free, what's the word for it? Whether COVID-19 escapes from the current vaccine, effectiveness of the current vaccine. Admittedly, it seems that there be two dimensions to it. The first is how much potential the COVID virus itself has to mutate into to escape. And on the second hand, how many cases of COVID there are out there now, giving it more of an opportunity to find what few there are. Mm-hmm. And having said that, just from the COVID-19, this is just what we know about the COVID-19 virus itself. what's its potential in terms of the structure of the virus for an escape compared to other viruses, SARS or some of the others around? Is it among the highest or lowest, or we don't know?
You know, my impression, this is just my like personal impression, is that I don't think it's like significantly more. I think influenza, the virus that I think mutates the most of the ones that we like are worried about in like current day is HIV. I think HIV just mutates so rapidly. It has such an error-prone kind of replication process that in a given individual, like you'll just have what are called swarms, or like, which are just like lots of different HIV variants in a given individual who's infected. Now, for SARS-CoV-2, I don't think it's as high. Like everybody is mutating to try to get to like a more effective point of survival. But I think that HIV really takes the cake in terms of viruses that are kind of circulating in kind of modern, like in our world right now, that we really need to think about mutation rate. HIV, this is actually one of the reasons why there, like HIV, there is no HIV vaccine. And this is in part of the reason is that it is so wildly mutagenic. And, you know, so what people are doing there in the setting of HIV is they're creating these kind of mosaic vaccines where they're like, you can't vaccinate with like what Moderna or Pfizer did, which is just like one sequence for the spike protein. So they're like figuring out like, what is a mosaic of like different HIV sequences that one could make, and like vaccinate with that to try to capture all of the like how much mutation is on the HIV protein or HIV will undergo. So so yeah, I don't think SARS-CoV-2 is, at least at present, given the kind of the evolution of how many mutations we've seen, given the number of people that have like had a case of SARS-CoV-2, I think my guess is if HIV had that many human hosts at present, we would see a lot more genetic diversity.
Just to follow up, do you think that we'll ever have an effective? Do you think we'll ever have an effective tuberculosis vaccine?
Yes, in fact, yes. I don't want to spoil the next talk that I'll give to you all probably in like hopefully a few years, but I'm actually very optimistic. So let me tell you why. So for tuberculosis, actually that ineffective vaccine that I was kind of critiquing earlier, BCG, it actually turns out really oddly that if you in non-human, so okay, I'll tell you I'll finish my sentence and then I'll tell you like my tempering of the good news. So if you actually just take that vaccine in non-human primates and change it from a subcutaneous or intradermal delivery, and you instead deliver it intravenously, it actually entirely protects monkeys from TB disease. Okay. And so what that suggests is like the route of vaccination matters. Now, okay, then we have to like, you know, temper ourselves with the practical ramifications of intravenous vaccination for 2 billion people. And there I become a little less optimistic just in terms of how easy it would be to deploy something like that. But what it does suggest is now we actually have a correlate of protection in the lungs, or like that we can actually now triage lots of vaccine candidates for. So what gives me hope is, you know, actually never in human history have we had, except for COVID, have we had so many vaccine platforms tested for the same pathogen all at once, right? So again, if any of you have friends in like Pfizer, Merck, any of these big pharma companies, tell them to talk to me, because really what I would love to be able to understand is you know, like now we have all these controlled studies in humans where like all these different platforms are being tested with basically the same protein, the spike protein, just delivered 17 different ways. And those are all being delivered. And then you can look at the immune response that's being elicited in people. And so if we have a hypothesis for like, here's the immune response that this intravenous BCG is generating, then we could say, just like, let's go to the menu of all of the vaccines that have been tested for COVID and ask which one generates the immune response that's most similar to that one. And then can we just like boot that up for a TB vaccine? So that's actually what gives me hope, is like, you know, I think if you're creative, you can think about, yes, okay, so for me as a molecular immunologist, like I am so excited, right? Like I look at this like, yes, these like last 15 months have been awful. I live by myself. Like I've, you know, I have a bunch of plants, right? Like I have not seen my grandparents for like 18 months. Like this has been rough, right? But, you know, if I think exclusively through the professional lens in which I live, like this is like, oh my gosh, it's like immunologist Christmas, because like we've never had this kind of data in humans, right? Because of all of the clinical trials that people had to do. So I was like, can I just like have access to those data? But then, you know, pharma companies don't actually want to give you the access, because nobody wants to know that their vaccine is slightly better or worse, right? Nobody wants to do the head-to-head, but I need it. So.
You know, full court press works. Get a lot of people involved, we can solve anything. Oh, well. Any other questions? I don't see any more in chat. Okay. Wow. It's been fantastic. I really enjoyed this talk. It was really great. I think everybody did. So if there are any more questions, I think we'll close out, but I want to thank you very much. And very much enjoyed it. And yeah, and we'll give you about 3 years, and then we'll call you back and see what's new, because boy, this field sure changed since, you know, when we were at MIT, a bunch of us back in the 60s, 70s, 80s, they didn't know anything about this stuff. And biology was a teeny tiny department, and MIT was electrical engineering and computer science and physics. So it was great to see how the place has changed, and I really enjoyed your talk.
Great. Awesome. Well, have a good Tuesday. Take care.
Very good. Excellent. Bye-bye.
Thank you again. Bye.